So let's move on to proteins, the third of our macromolecules. Proteins are made up of amino acids, and you may have seen some of this before in terms of your 20 different amino acids. Basically, amino acids have a carbon, hydrogen, oxygen, and nitrogen, and then sometimes they have a sulfur and a phosphate group. So depending on what this group is up here, their carboxyl group, is going to determine what type of amino acid they are. But the same thing is true as it is for all of our other organic compounds. If you take away water, you dehydrate two amino acids, they will form a bond, and this is called a dipeptide, meaning two peptides. And it's a dipeptide bond or a peptide bond. That's important for you to know. If you wanted to break up a dipeptide into its amino acid components, then you would just add water. So some examples of the different types of amino acids, because of their different carboxyl group, includes glycine, which is the simplest of all the amino acids. Aspartic acid, you can see that it has a larger carboxyl group. Then we have lysine, which has an even larger one. And then we have cysteine here, and then there are several more. Protein structure is important because it dictates how these amino acids, once they're bound by peptide bonds, can make different structures. So we have what we call the primary structure, and that is just a bunch of amino acids bound together by peptide bonds. One nice flat straight chain. What happens is that they will start to coil around or fold, and this is when we get a secondary structure. So a primary structure will give way to a secondary structure when it starts to kink and spiral. This is what we see in DNA. But then you can get even further folding or kinking, so to speak, of these proteins, and then we get what we call the tertiary structure. And then the tertiary structure can fold even more, forming the quaternary structure. And an example of the quaternary structure is a hemoglobin molecule. So it has a quaternary structure. Again, this is just another schematic to show you the different levels of structure of how proteins can arrange themselves. So a simple protein chain of amino acids, polypeptide, is the primary structure. Then it can fold into the secondary structure, which is what we see here in the helix, as well as the pleated sheet. And then it folds more to form the tertiary structure. And the tertiary structure will fold even more to form the quaternary structure. There are two types of proteins. We have fibrous proteins, which are involved in structures, and globular proteins, which are involved in function. Proteins are probably the most important macromolecule in the sense that they provide us with structure and they do so many things. Fibrous proteins are strand-like. Here on the left, collagen is the most common. They're water insoluble and they're very stable. Keratin is an example, like in your nails and your teeth, elastin, collagen, and some contractile fibers. Globular functional proteins are more, like I said, functional. They're compact, they're spherical, they are more water soluble instead of insoluble, and they're sensitive to environmental changes. They also tend to have active sites, and a good example is hemoglobin in our blood that binds oxygen and carries it. So some other examples are antibodies, hormones, molecular chaperones, and enzymes. Molecular chaperones would be like what we just talked about that gets the lipids through the blood because they're hydrophobic. Another thing that's really important about proteins is that they can be denatured if the temperature gets too high. So for instance, when you have a fever, fevers are not always bad. Fevers are good. They can kill viruses and bacteria that are infecting you and invading your body. But in some cases, you don't want a temperature, and we don't want it to get over 104, really, because you start to break down these proteins and their structures. So protein denaturation can occur with an increased temperature or a change in pH. And this is one of the many reasons why we have a range that we want to keep these things in. And in some cases, protein denaturation, where it falls apart or comes apart, can be reversible if the normal conditions are restored, but sometimes it's not. Sometimes it's irreversible. So, for instance, when you're cooking an egg, those are proteins that when you provide enough heat to that egg, it changes its form. It's denatured beyond repair.